Hello, everyone. Thank you all for coming to today's seminar. It is my pleasure to introduce today's speaker, Professor Ron Naaman, who is joining us from Israel right now. Ron is a professor of chemical physics at the Weizmann Institute. During his time at Weizmann, he has also served many other positions, including the chair of the Institute's Chemical Services Unit, the head of the Department of Chemical Physics, as well as the chair of the Scientific Council at the Institute. Ron's research focuses on electron interaction with molecules, and in particular, chiral molecules. His group studies spin-dependent transmission of these molecules, as well as explore importance of electron spin in various bi-related electron transfer systems. It is our pleasure to have Ron here with us today. And before I turn it over to Ron to learn more about his interesting work, I just want to remind our participants to please keep the questions to the end of the talk. You can either use the Q&A feature of Zoom to type your question, and then we can ask the question for you, or you can raise your hand and directly ask your questions. So with that, Ron, thank you very much for being here today, and I'll turn it over to you.
Thank you and good afternoon. Thank you for inviting me. It's a pleasure to give a talk, although from such a distance. My talk is concentrated on two features, the electron spin and chiral systems. And I would like to provide you with some new angle on these two subjects. So the players are chiral molecules. In chemistry, chiral molecules are well known, mainly because their importance in biology. We know that nature is built from chiral molecules. But usually when we relate to chiral molecules, we relate to their structure. And one does not associate with chiral molecules any special electronic properties. The second player in this talk is the spin. Again, we know a lot about spin. However, in chemistry, typically we relate to the spin specifically when we have either ferromagnetic or paramagnetic molecules. We don't think a spin is an important property in a closed shell system when all the electrons are paired. What we found, and that will be the subject of my talk, that chiral molecules, although they typically closed shell, they have special electronic and spintronic properties that may provide us some answers on really important questions in nature, and I will elaborate on it in a second. So what did we discover? We found that when electron is transmitted through chiral potential, and here you have an example of a chiral potential, like electron sliding down a slide. Here it's more presented in a more physical way. We realize here, even taking this classical picture, that there is a force that must act on the electron that moves through chiral potential, and this force is perpendicular to the velocity of the electron. It is the classical centripetal force. However, in the rest frame of the electron, this force is exactly like effective magnetic field acting along the molecule. So we realize that by the very simple fact that electron has to go through curvatures in a chiral system, it feels an effective magnetic field. And this effective magnetic field, of course, can interact with the electron spin that has a magnetic dipole. And as a result, it creates a spin-orbit coupling here that's amount to this effective field times the magnetic dipole of the spin. So when electron is going through the system, one spin gets stabilized, the other gets destabilized. Which spin is stabilized and which spin is destabilized depends on the velocity of the electron. Of course, it depends on the electric field. And electric field is basically the handedness of the molecule, because you realize that for one handedness, this centripetal force will work if on the left if for the electron moving down, while for the other, for another handedness, it will act from the right of side of the electron. So the sign of the electric field is defined by the enantiomer. So now we realize that if one spin is stabilized going down, the opposite spin will be stabilized going up. And that's an important feature here, that the spin and the linear momentum of the electrons get coupled. This is a property that in physics people know it and know about it, and it's related to topological materials. There are many theoretical work that were performed on this system, and usually they suffer from the problem that the effective spin polarization they obtain using this model is too small, and I will touch it again in a few minutes. So how we measure this spin polarization? A simple setup is one in which we have here a magnetic substrate. We can magnetize it either pointing up or pointing down. We adsorb molecule on it, for example, DNA. Then we come with a conducting AFM, with the tip of conducting AFM, and we measure the current going through the DNA in this case, or for other molecules, as a function of the voltage applied, as a function of the direction of the magnetic field. And here I present the results obtained for double-stranded DNA, different lengths, 20 base pair, 30 and 40 base pair. And you see clearly that we get different IV curves for the two magnetic direction. When we put it on a log scale, we see immediately two effects. One, that there is different threshold for conduction for the two spins. And secondly, that the conduction, the current doesn't depend linearly on the voltage. And that's an important finding. That's what allows us actually to measure spin polarization in two-point contact. And here I present the results for double-strand DNA and for alpha helix oligopeptides of polyalanine. And you realize that in principle, the spin polarization scales with the length of the molecules. Now, spin polarization is defined as the current measured for one spin minus the current measured for the opposite spin over the sum. So when we speak about 60% spin polarization, it means that the ratio between the currents is about 4:1. So we get relatively high polarization at room temperature with molecules that are very short. You see here only 3 nanometer in the case of oligopeptide and DNA, even if we get to 12 nanometer and we get 65, close to 65% spin polarization. A very efficient spin polarization at room temperature. What is interesting is to look on the length dependent for each spin independently. So what you see here is the current, or log of the current, as a function of the length of the DNA molecule. And you realize for one spin, the preferred spin, we see very weak decay of the current as a function of distance. The other spin decays much faster. That we can understand by the fact that the spin and the linear momentum are coupled. Because they are coupled, the preferred spin cannot scatter back without flipping the spin to the unpreferred one, and that is highly improbable, and therefore the backscattering is suppressed here. In the case of the wrong spin, there is large backscattering, and therefore the conduction decays very fast. So to summarize what we observed so far, we see that the current versus voltage curves are not linear. There is a different threshold for each spin conduction. This is an indication that spin doesn't flip during the conduction. Namely, the filters, the spin filters, the molecules, the chiral molecules, are passive filters. They don't actively flip the spin, they only allow one spin to pass and block the other spin. The polarization is similar to the polarization obtained for the same molecules when we transmitted photoelectrons from them, namely unbound electrons. And that is quite surprising that we get the same spin polarization independent if the electrons are bound or unbound when they go through the system. Again, we see the coupling between the linear momentum and the spin, and that's, we can realize it's a reason for long-range electron transfer. Perhaps that explains why in biology electrons are transferred through chiral molecules, through proteins that are chiral, although they are not good conduction, but they are chiral, and therefore backscattering inside the protein is suppressed. Now, in order to check what other properties of the molecule affect the polarization, we joined forces with the group of Bert Meyer from Eindhoven. And Bert is expert in making supramolecular structures made from this type of molecules, porphyrins and so on. And what is interesting here, the main molecule is not chiral, but by putting side groups that are chiral, he is able to induce formation of wires that are chiral. Namely, if the side groups have one L chirality, then the supramolecular structure will be a left-ended chirality, and if the side groups are right-ended, then we see right-ended chirality here. And if the side groups are achiral, then it makes the supramolecular structure random in terms of chirality. So we have these wires, and we put them on the surface. So you see this wire lying on the surface. Now we use our AFM that I just described, and we measure the conduction through these wires, namely through the diameter of the wire. You should realize the wires are lying. We measure it perpendicular to the long axis of the wire. And here it's the height. So that's the diameter of the wires, and typically between 15 to 20 nanometer. Namely, these wires are not single, they are not made from single molecules, but there are many molecules at the diameter. So now I'm going to show you the IV curves obtained for these wires for different molecules that Bert Meyer provided us. And depending on the molecule, you see different polarization, spin polarization. You realize that for some molecule, we get spin polarization as high as 90%. Huge spin polarization. So that's by itself is interesting. Actually, we understand the difference between this molecule and this molecule. Here, the wire is closer packed than here. So clearly, the spin polarization is not a local property of a single molecule, but it's a property of the wire. And to prove it, we did the following. This, in a supramolecular chemistry, people use what is called surgeon and soldier. What it means, they mix some chiral molecules with achiral molecules and induce the formation of supramolecular structure. And what is found that if you dope the achiral molecules with some chiral molecules, the chiral molecules induce a chirality on all the supramolecular structure. So you see here the CD spectrum, that's the intensity of the peak of the circular dichroism spectrum. And you realize in the beginning, when you add chiral molecules, surgeons are the chiral molecules, you add them to the soldiers, to the achiral molecules, the CD spectrum, the optical activity depends linearly on the amount of chiral molecules. But here we see change and we get amplification. So the chiral molecules induce chirality beyond their own structure. And this effect is known for a long time. Now we took for the same molecules that were measured here, we went and we measured their spin polarization. And here is what we found. We found that the spin polarization through these molecules follow basically in an amazing way the intensity of the peak in the CD spectrum. That's very impressive. What it tells us again, that there is a relation between a global property of the molecule, of the wire, its optical activity, and the spin polarization. So with the models I presented before, there are several problems, and I want to summarize them quickly. First of all, typically they come with relatively small spin polarization. So the spin polarization they find in the models that I just presented before are on the order of few percent, while as you saw, we are obtaining, we and many other groups in the world now, obtaining spin polarization of many tens of percent up to 90, and somebody even got 100% spin polarization. They do not, these models that exist do not relate the spin polarization of photoelectron to that of the bound electrons. So it's not clear how they related according to this model. There is no relation to optical activity, and therefore we propose a modification of the model, which is combination of the spin orbit coupling that I just showed you and spin blockade. And I'm not going to show you all the formula and so on, only I will give you a simple scheme of what's going on. When you have a chiral molecule, let's assume it's between two leads. It can be also between donor and acceptor. It's the same phenomenon. Now we applied voltage. When we apply voltage on a molecule between two leads, what happens? There is a charge polarization. Namely, charge start to move in the molecule and a dipole moment is created. This charge motion, because it means that electrons are moving in chiral potential, involves some spin polarization. Very small spin polarization, because the spin orbit coupling, as I mentioned, is relatively small. So let's assume it's about 3% spin polarization, far below what we measure. But now when another electron introduced into the molecule, this electron penetrating to the molecule feels the spin that was left behind. So only electron with the spin opposite to the spin that was left behind, only this electron can penetrate in. For introducing an electron with the same spin than the spin that was left behind, we have to put extra energy. And the extra energy is the spin polarization. Let's say it's 3% or so, and times this exchange the spin exchange interaction, which is basically the splitting between singlet and triplet. It's on the order of 1 electron volt. So even for only 3% spin polarization due to spin orbit coupling, we get enhancement due to this spin blockade, and the difference in barrier now is 30 milli-electron volt. Large and huge enhancement. So it's important. This model explains first why the conduction is non-linear, because here you see there are two effects of the voltage. One, polarizing the molecules. The second one, introducing the electrons from the anode. The second effect is that this polarization here is related to the anisotropy of the electrical polarizability of the molecule. This is exactly the same term that contribute to the optical activity of the molecule. And that's how the spin polarization is now related to the optical activity. If you want to read more about it, there is a feature article in Journal of Physical Chemistry Letter that we published just not too long ago. So what does this model provide? First of all, we get large different barrier for injecting one spin versus the other due to the spin exchange interaction, namely spin blockade. The spin polarization depends on the anisotropic polarizability, as I mentioned, like the optical activity, and this provides us the connection between CISS effect and optical activity, and there is no need for coherency in the conduction, and that's something that was observed by many, that actually the conduction doesn't have to be coherent in order to obtain the spin polarization. Now I will present to you some applications of the CISS effect, and the first is Spintronics devices. And I know that in the Nano Center, Spintronics is a big issue. So typical Spintronics device is a spin valve. And as you know, it's already commercial. All of us, we have spin valve in our reading head of the hard disk. And basically, what happens in giant magnetoresistance, there are two ferromagnetic layers with a non-magnetic layer in between. And if the two ferromagnetic layers are magnetized parallel to each other, we get low resistance. If they are anti-parallel, we get high resistance. And therefore, what we see that the resistance depends in a symmetric way on the magnetic field. Doesn't matter if these two magnets pointing up or down together. In both cases, we get low resistance. We are going to present CISS-based spin valve. So what is the idea here? Here we use chiral molecules. So instead of one ferromagnet, we use chiral molecules. These chiral molecules polarizes the spin. And then we have only one ferromagnetic layer that serves to read it. And so what we expect here, that if the ferromagnet here is pointing so that the electrons coming polarized from the chiral molecules can penetrate, will get low resistance. However, if the ferromagnet is pointing the opposite direction, we expect to get high resistance. So we expect to get non-symmetric dependence of the resistance on the magnetic field. And sure enough, here it's the work we did with helicine together with Jean Lacoeur from Geneva. And what you see for helicine, for P helicine, we get this shape. For M helicine, we get the opposite shape, and we get nice magnetoresistance. What is interesting, it's basically this magnetoresistance doesn't depend on the temperature, and it's stable for all temperature. It can stay like this forever. It is very robust because actually the electrons going through the helicin go basically elastically, and they don't deposit energy in the helicin. Now, what we can do is something even more interesting. Instead of using chiral organic molecules, we can use chiral oxide. There is a method that was developed, among others, with Yitzhak Mastai from the Bar-Ilan University. And following this method, we make aluminum oxide, chiral aluminum oxide. In the meantime, we did also chiral copper oxide and chiral cobalt oxide. And here we use this chiral oxide layer, and we get huge magnetoresistance at room temperature, and it's very robust. So here we have a method that can be used actually using conventional clean room facilities with no need for any wet process. Now I want to discuss the issue of what happens in chiral molecules when we apply field on them. You know, typically electric field and magnetic fields are not strongly related in terms of technology. It's very difficult to find a method to use electric field to get magnetic field. Chiral molecules are a good candidate to do it in a simple way. So what we have here, we took short polymers that's were done together with a good group from Kanazawa in Japan. And we have to take a polymer. The polymer can be either L or D polymer. And we deposit the polymer on this device. And now, what is this device? It's a Hall device. You probably know what's a Hall device. Basically, it's made from, in this case, gallium nitride, aluminum gallium nitride, with 2d electron gas in between. And we have two electrodes, source and drain. Current is flowing here. And if there is typically in the Hall device, if there is a magnetic field applied perpendicular to the surface, then we get potential perpendicular to the current flowing. In our case, we don't apply magnetic field. Instead, we adsorb here chiral molecules. And on the top, we put a gate electrode. So what we do now, we apply voltage between the gate and the device. The voltage charge polarize the chiral molecules. And what we find when we measure the Hall signal, we get Hall signal that's for L and that's for D molecule. And you realize that we get the Hall signal, the direction of the signal depends on the chirality, because we inject into the Hall either one spin or the opposite spin, and here you see the result. So what we have here is the following. When we apply electric field along the molecule, charge is moving. Because charge is moving in chiral molecule, we get spin polarized associated with each electric pole. Now, it's a transient effect. It's not an equilibrium effect. Namely, when we apply the field, we get the spins here, the opposite spin here. So we are still singlet. The total spin here is still 0, but at each pole, we have spin polarization. Now, which spin associated with which electric pole depends on the handedness of the molecule. I want to make a point here regarding this effect. You know, we start with a singlet. As you know very well, in singlet, we have two electrons that are paired. They are entangled. The wave function is alpha beta minus beta alpha. So we now apply electric field. We can also excite a donor on this side. It's the same thing. An electron, because of the field, is now moving to this side. When electron is moving to this side, one spin is preferred. That's exactly like measuring the spin of the electron. So what's happening here, this electron transfer breaks the entanglement. So now we have here an electron that's either alpha or beta, depending on the handiness of the molecule, but it's not entangled to the electron left behind here. It's maybe correlated, but it's not entangled. That's an interesting feature of chirality. Another interesting feature is that the electron here is coupled to the molecular frame. You know, the spin when we have unpaired electron in acyl molecule, the spin is not coupled to the molecular frame in a significant way. That's why in ESR we apply magnetic field, and the spin is aligned according to the magnetic field, but not according to the orientation of the molecule. That's not true here. Here, the spin is coupled because of this strong spin-orbit coupling and exchange interaction that I mentioned before. The spin is coupled to the molecular frame. And in the moment you will see that it has very interesting results. So what it means now, think about two chiral molecules that are coming to interact. Very schematically, let's assume that what we have here is two molecules that are not charged. Now they come nearby each other, due to induced dipole, two dipoles are formed. As I mentioned, when we have dipoles, associated with this dipole, there is spin polarization. So when the molecules interact here, we realize that if they are the same chirality, we get here singlet. If they are opposite chirality, we get triplet. So we get here a term in the interaction that has to do with the chirality of the molecule, the enantio-selectivity of the molecule. So what we indicate here, that enantio recognition, it's not only lock and key type effect, it has to do also with the electronic term, new electronic term in the interaction. And to prove it, here's the experiment that was done together with Mital Reches and Yossi Paltiel from the Hebrew University. And I'm summarizing briefly here. We put on the tip of an AFM chiral molecules. The chiral molecules are absorbed from the surface. And basically, we measure the force between the tip and the surface when the two molecules are homochiral or heterochiral. And what we find, look here, you see clearly that if they are, in one case, if they are the same enantiomer, we have this binding energy, it's the force here that we have to apply, and if they are heterochiral, the binding is, it's much weaker. And here you see the difference, and the difference we are speaking here it's given in terms of force. In milli-electron volt, we are speaking on between 100 and 200 milli-electron volt. And here, some calculations that were performed by the group of Sasson Shaik from the Hebrew University. And what you realize, what we plot here is the different energy between homochiral and heterochiral. And you see different energy is on the order of several kilocalories. Depends on the distance, of course, but even at this long distance of about 3 angstrom, we still have about KT or perhaps even be above KT difference in energy. And what is very interesting that the difference is depends strongly on the orientation of the molecule. So what it tells us that in chiral molecules, the reaction become more angle dependent than for achiral molecule, and it's only because the spin is coupled to the molecular frame. Only to demonstrate to you the amount of the energy involved in this process, here we absorbed chiral molecules on a ferromagnet that was initially not magnetized. And there is a pattern here, the oligopeptides that were absorbed on the ferromagnet. And now that's the topography, and you see clearly the squares of the two molecules. Now we came with a magnetic FM tip and measure the magnetic force. And here is what we get. We get for the L enantiomer, we get the magnetism pointing up. For the D, it's pointing down. And why it's this? Again, it's the issue of the spin associated with the induced dipole in the molecule, where the molecule has to bind to the surface. Depends what spin is associated with the pole pointing towards the surface. And in order to bind it and to gain the free energy associated with this binding, it's worth for the ferromagnet to flip the spin to the right direction. So in this case, we get binding. If it would remain in this orientation, we'll not get binding. So we have, sorry. So we see here clearly that we have we induce the magnetization in the ferromagnet by the adsorption of chiral molecules, and the handedness of the chiral molecules define the direction of adsorption. And this can be used to separate an enantiomer, because if we have a ferromagnet oriented already in one direction, it will prefer to interact with one enantiomer versus the other. And here is the demonstration. We take ferromagnet, we adsorb it L molecules. You see they adsorb very efficiently. On the opposite direction of the magnet, much less. And with D, it's the opposite. And here you see the ratios. And clearly, the effect is not a magnetic field effect. Here we applied magnetic field without having magnetized surface, and there is no effect. The all effect comes from the spins in the substrate. So here we have a way to separate an enantiomer using ferromagnet. And here you see the rate of adsorption of DNA on ferromagnet. And again, the rate depends on the direction of magnetization of the ferromagnet. But we can go one step further and check if we can use this method to separate an enantiomer by crystallization. You all know that Pasteur crystallized racemic mixture, and then he took tweezer and separated the enantiomer by looking in the microscope. We are in the Middle East, we don't like to work so hard, so we found a different way to do it. We simply put a magnet. And as you will see in a second, one enantiomer tend to crystallize more on magnet pointing north. The other enantiomer prefer the magnet pointing south. And here you see for glutamic acid, the colors are false colors, of course. The magnets are here. It's interesting because crystals start to be formed here, but then the system, some small crystal diffuse and they become larger, and then they fall down on this half. So basically, one half is enriched with one enantiomer and the other half is enriched with another enantiomer. And here it's another device that we made and allows us to get very high enantio excess in crystallization without the need to seed. Usually today, in order to separate enantiomer by seeding, one use enantiomer by crystallization, one use seeding. We don't have to seed anything. We simply put magnet and we get the crystals. And this provides us with a generic method for enantiomer separation. And the last part that I want to mention very briefly, it's the issue of artificial water splitting. You know, in water splitting, we try to get hydrogen from water. It's a 4-electron process. There are two problems in artificial water splitting. One, typically one needs potential of about 0.6 volt higher than the thermodynamic limit, which is about 1.2 volt. And secondly, we form byproduct, hydrogen peroxide, that not only reduces the amount of hydrogen we produce, but also destroys the cell. So the question, how we overcome this? So to understand it, let's go and see what happened to the spins here. In the case of this reaction, OH minus donate electron to the anode and form OH radicals. And we have two OH radicals that is now to react. Each OH radical is unpaired electron. And if these unpaired electrons are counter-aligned, namely they are on a singlet surface, then we will form hydrogen peroxide, which is singlet. However, and we will not be able to form the oxygen that we want, which is triplet. Now, if we align the spins and they are parallel to each other, we are on a triplet surface, and therefore we will not form hydrogen peroxide, but we will form oxygen and hydrogen as we want. So the whole idea is, let's make all the spin be in a line, and the way to do it is to cover the electrode with chiral molecules, and specifically in our case, one example, we use many systems by now, but one example that I present here is iron oxide coated either with chiral or achiral molecules. What is interesting with iron oxide nanoparticle, when you cover it with chiral molecules, you see this spectrum in the absorption region of the iron oxide itself here. So there is induced chirality, if you wish, in the nanoparticle. This phenomenon is known very well, and we use it here. And here are the results. You see we have current density. When the nanoparticles are covered with chiral molecules, we get much higher current than with achiral. Here's the hydrogen production. Again, with chiral nanoparticles, we get much higher hydrogen production than with achiral one. And here we measure the production of hydrogen peroxide. we put some material that becomes yellow, indicator that becomes yellow when hydrogen peroxide is formed, and we measure the absorption. So in case of achiral nanoparticles, you see we get a peak here. However, when we have chiral nanoparticles coated with a small chiral molecules, short chiral molecules, we get reduced hydrogen peroxide production, and if we put longer molecules, we get even less hydrogen peroxide production. You see here, it's very, very low hydrogen peroxide production. So clearly we get both enhancement of the hydrogen production and reduction in hydrogen peroxide production. The same effect was observed recently using magnetic surfaces, and actually it makes this concept of using spin to control water splitting almost ready to be commercialized. In principle, what we do here, we make the artificial process closely related to the what happens in biology, because in biology, indeed, the molecules are chiral, and therefore we show that the spin is polarized there. And what is interesting in recent work, we and others showed that instead of chiral molecules, one can use chiral oxide, as I mentioned before, and chiral oxide are easy to make, they are cheap, and they serve very well in this process. So to summarize, the CISS effect in physics, chiral molecules are interesting nano devices. They produce spin polarized electron without the need for ferromagnetism. They introduces chiral molecules as a new topological material. In biology, this effect explains efficient and dissipation-less long-range electron transfer. We recently, together with the group of Moel Nagar from USC, we showed the spin polarization in some of the proteins that are long-range electron transfer medium, and clearly spin polarization exists there. And also in biology, our result may explain the efficient enantio recognition nature. In chemistry, we enhance multiple electron reaction, enable efficient enantio separation, and allows for spin enantio-selective chemistry. This subject I didn't discuss today, and if you are interested, there is a feature article in PCCP that summarizes it. Here are the group that does the work. Those people are former group member. Those are the present group member. I own them everything. They are really very dedicated group. And there are many collaborators, which I really appreciate, both for being great scientists and such good friends. And thank you very much.
Thank you, Ron, very much for a wonderful talk. We have a few minutes for a couple of questions, so please feel free to type your questions in the Q&A or raise your hand and you can directly ask your question. So let me start with the first question. So the question says that your model on slide 12 suggests the spin selection mechanism in which the energy potential barrier for injecting one type of spin is significantly different from the potential barrier experienced by the electrons with the other spin. The ratio of the injected electrons with different spins should depend on the temperature of the system. Yet in slide 16, you indicate that there's small dependence on temperature. How come? Is there spin scrambling upon the injection due to some scattering process?
Okay, it's a very good question. First of all, what we find that although I mentioned 30 milli-electron volt as a barrier, I used simple numbers, but actually, as you saw also in the calculation, what we find typically that the difference in barrier is more like 100 milli-electron volt. And therefore, the temperature effect in the range that we checked is very minimal. It's not a big effect. We find, I must be careful with the issue of temperature. We found systems in which the temperature plays an important role. It's not always correct that temperature doesn't matter. First of all, temperature may affect the structure of the molecule, and the structure of the molecule may affect the spin polarization. So it's not a general rule that there is no temperature dependent, but in many cases, the temperature dependent in the range that we study between room temperatures, about 100 K, is not significant.
Great. So the second question is asking that in the example on slide 19, the vertically standing chiral molecule is sandwiched between two horizontal electrodes. Could you comment on how the top electrode is deposited and whether upon application of voltage and injection of charge, there is any distortion in the shape or length of the molecule?
Okay, the way we make it typically, we make a close-spec monolayer, on top of which we adsorb, we coat about 3 nanometers of either magnesium oxide, typically magnesium oxide, that's what we found as being the best material, on top of which we deposit the top electrode. So that's the way we do it. And with the 3 nanometer magnesium oxide, the efficiency of making the devices is quite high. So it's working quite well. Regarding the structure of the molecules and so on, what we find that the devices are working without any modification in terms of their properties for a very long time. Because of the oxide layer on top, the molecules are quite protected from humidity, and as a result, the devices are stable for a very, very long time.
Great. So the next question is asking, I think your effect is unique relative to topological physics. You couple the spin onto the linear momentum direction and not perpendicular to it, as will be the case in topological insulators and Rashba physics.
Okay, one has to be careful. The spin orbit coupling direction is perpendicular to the velocity. So that's, in that sense, we have the same structure. Actually, when you go through the Hamiltonian, and if you wish, you can look in the paper published not so long ago in Journal of Physical Chemistry. See, one second, I will find it. And there you can see the Hamiltonian and everything with all the details, and realize, okay, here. And so this work with Karen Michaeli, this paper, you can find there the old expression, everything, and it is a whale. Basically, it's like a whale material in terms of topology. So the angular momentum and the linear momentum are coupled like in whale material.
Great. So the next question is asking, are there applications for the charge and spin polarization for characterization or stabilization of membrane proteins such as GPCR or other membrane proteins? Could the charge or magnetic field be manipulated to get the orientation of the molecule correctly on the surface, such as nanoporous chips, for either at the affinity capture kinetics or in line with a mass spectrometer?
So, okay, it's a far-reaching question. Part of it we did. We used electric field to induce a selective adsorption on surfaces of oligopeptides. This paper was published last year, and definitely it allows to control this connection between electric field and magnetic field, allows to control the adsorption in a very interesting way. Now we believe that also in terms of binding, we have proof now that if we add to a protein molecules with the right chirality, the molecule can contribute charge into the protein. While if we put it in the wrong chirality, the charge is not penetrating into the protein. So that's another way to control activity of proteins by attaching molecules with one enantiomer or another enantiomer. The effect is not simply structure, it's really the injection of the charge. And there is a paper now under review on this subject.
Great. So can you use this spin selectivity and breaking of entanglement to facilitate singlet fission in some way where this might be desirable?
Okay, we only start now to explore this issue. We have several experiments now with EPR to try to control first of all the spin, to see if we can manipulate the spin by injecting a spin polarized electron into already polarized spin and so on. Yeah, in principle, we can do it. We have scheme to do it, but we didn't do it yet. So it's a challenge.
So more on the barriers for spin injection. You mentioned the singlet triplet gap, but I think you were saying that it is easier to inject the spin of the sign of the spin that is depleted. I understand this, like the Pauli principle, if you're thinking that the spin polarization creates an empty state. So a ground state model, is this correct? If you injected a hot electron into the LUMO excited state, could you see the opposite injection barrier, wherein the triplet energy would be the lower energy state?
Okay, yeah. First of all, it's exactly the Hund's case, and it's what the person that asked the question is completely right. We are able to inject the opposite spin in the excited state and so on. All this is true, yeah.
Great. So we have two more questions. How generalizable is your new method for separation of enantiomers? Have you tried some molecules more complex than simple amino acids, for example, drugs?
Yeah, the answer is positive. It's very general. We still have to prove the efficiency with systems that have more than one chiral center. But for one chiral center, the effect is very general, and we separate many molecules by now, not only amino acids. There is a commercial attempt to generalize it, and I hope it will work.
And the last question says that it seems that most chiral organic molecules featured today could be classified as insulators in reference to their bulk electrical conductivity. Are there challenges in observation of these effects across longer length scales in organic materials with greater bulk electrical conductivity?
Okay, here I want to say something. I didn't stress it in a clear way. You may wonder why nature transfers charge through proteins and not through conjugated molecules. You know, even if you take the photosystem, there are conjugated molecules there, but the charge is transferred through the insulator, so-called, the protein, and the conjugated system is used only as antenna. So why so? So I believe the answer is that what is important for nature is control over the transmission. It's really not the amount of charge you transfer, but for nature, it's important that every electron that starts to move will get to the end and not will get stuck in the middle and will charge the protein. This you do with insulator, chiral insulator, better than you do with a conductor. And I believe that's the reason that proteins are utilized by nature so persistently for charge transfer and never a conjugated molecule, at least never that I know. So it is a really interesting issue that what's important for nature is not the current, not the amount of charge that's going. What's important for nature is the fact that every electron that will penetrate into the protein will get to the end. And that says something about those that try to mimic electronics from biology. We have to remember, not for all electronic applications biology will fit, because in biology, nature didn't optimize for current. It's really important to understand it. So I think if you like nature, for nature, what is important is efficiency, not the current.
Great. So there's one last minute question. I'll ask this, and this would be the last one. Is there any role for external time-varying magnetic field in your study?
Okay, so it's important to appreciate that the internal field, this effective field that I mentioned, if you calculate it, it's thousands of Tesla. So external magnetic field doesn't affect the electron while passing through the molecule, chiral molecule, because the internal field is huge. The only way to control with magnetic field is to control the electrons. Then you can either accept the electron or make a barrier for this spin. But the CISS effect itself is quite robust against magnetic field because the internal magnetic field is huge. And for those of you that know a little bit about graphene, you may remember that when you bend graphene, people observe very large spin orbit coupling, and the effective field that they get for bending graphene is hundreds of Tesla. So it's very similar to what we observe here, that chiral molecules, because the curvature that creates effective magnetic field, which is thousands of Tesla.
Great. So those were all our questions. Thank you, Ron, very much for a great talk and for being here today with us. We really enjoyed the talk. Thank you.
Thank you. Bye.
Bye. Thank you all for attending the seminar today.